Hey guys, this is Tanner, Maddie, and Braden. Working on week 7 group problems. So we'll let Maddie go ahead and get started.
Oh, I'm coming up here. Okay. So we're being really ghetto and just using our notes today because it's too much effort. So for the first one, it gave us 4 different genomes, percentages of different base pairs within each sequence. And we were supposed to figure out if it's RNA or DNA, double-stranded, single-stranded, circular, or linear. So for the first one, we said it was RNA, double-stranded, and circular. So it was RNA because it has U. It has actual U in their sequence. And then it's double-stranded because there is for like the A to U and for C to G ratios, they're super similar with their percentages, which is normally common with double-stranded sequences. And then we said it was circular because it was resistant to exonucleases. For genome 2, we said RNA, single-stranded, and linear. So RNA because of the U, single-stranded because the ratios between the different groups aren't very close, and linear because it's sensitive. Genome virus 3, we said that it was DNA, double-stranded, linear. So DNA because it has the T, not the U. Double-stranded, again, because the ratios are similar, and linear because it's sensitive. And then for virus 4, we said that it was DNA because of the T, single-stranded because of the ratios, again, and circular because of the resistance. And then I'm just going to go through number 2. You do have that down. Okay. So A, topoisomerase 2 is composed of 4 protein subunits and uses ATP to compact bacterial chromosomes, while a nucleosome contains 146 to 147 base pairs of DNA and 8 histone proteins. B, the DNA double helix is 2 nanometers wide. Adjacent base pairs are separated by 0.34 nanometers in the DNA double helix. C, topoisomerase 1 introduces positive supercoiling into DNA and cuts 1 DNA strand. D, the origin of replication in E. coli is called ORIC. E, in the Hershey-Chase experiment, bacteriophage T2 proteins were labeled with a radioactive isotope of 35S, while bacteriophage T2 DNA was labeled with a radioactive isotope of 32P. And lastly, the sugar in uridine is ribose, while the sugar in deoxythymidine is 2-deoxyribose. And all of those answers can be found in the readings.
All right, sweet. So for number 3, let me go to it real quick. How I use your computer. Scroll down. Okay. So for number 3, for A, how long is the DNA portion of this translocation? Since both chromosomes were hooked together in their entireties, we added the 48 million and the 109 million together. And then since I like working with picometers, I times that by 340. And then to get that down to meters, I just converted it. So I got that down to 0.05338 meters. And then for B, to find the nucleosomes in that, there's 146 base pairs per nucleosome. And since we know that the linker DNA is 50 base pairs, I just added the 2 together. So what we did is we took 157 million and divided it by 196, and we got 801,020 nucleosomes total. And then for C, we know that there's 2 H2A molecules per nucleosome, so we just multiplied that by 2 and got 1,602,040 H2A molecules. And then D, we just found a range of the radial loops because we read that it could be anywhere from 2,500 base pairs to 200,000 base pairs. So on the bottom part of that range, we did 157 million divided by 200,000. We got 785 loops. And then on the upper side, 157 million divided by 2,500 loops to get, or divided by 2,500 base pairs to get 62,800 loops. So our range for that would be 785 to 62,800 loops. And then for number 4, it was kind of similar. So for A, we know there was 4.4 million base pairs. So again, I like picometers, so I just used picometers. I times that by 340 picometers and converted it. We got 1,499.4 micrometers. And then B, if we connected that together to get the circumference, we got, well, the 1,499.4 micrometers, then we just divided that by pi to get the diameter. So the diameter of that, we got to be 477.2 micrometers.
All right. Moving on to 4C. It's wanting to know how, and with one of the loop domains, which contains 50,000 base pairs, which one of these, if it was stretched out linearly, how long would it be in micrometers? So similar to part A, you take the 50,000 base pairs. Also, I like working with picometers and multiply it by 340 picometers, and then converting from picometers to micrometers by multiplying by 10 to the negative 6. That gives you a total of about 17 micrometers long. So one loop domain would be about 17 micrometers long. And then for D, it says how many histones are found attached to the M tuberculosis chromosome. As we started working this out, we realized that histones aren't present in bacteria, only in eukaryotes. And so the answer to that is 0. Then for 5, okay, so for 5, want to know which of those 6 pictures were nucleosides. So what you're looking for here is basically a nucleotide, but without the phosphate groups. So that gave us A, E, and F. Those images didn't have the phosphates. The nucleotides, it's the nucleosides with the phosphate group, and those are the remaining ones, B, C, and D. And then for part C, it wanted to know which ones contain ribose. You just figure that by looking at the sugar cyclic rings. And the ones that contain ribose are C, E, and F. And the ones that contain deoxyribose are A, B, and D. And then finally, it wants to know which ones contain pyrimidines. And basically, all you're looking for that is in the nitrogenous bases, whichever ones only contained 1 cyclic ring as opposed to 2. And that was C, D, E, and F. So yeah, that is all the answers. Just go ahead and post that there for you. Thank you for another session with Tanner, Maddie, and Braden.